Hello guys, my name is Purine Sarkar and welcome to Bioscience World. So today I'm going to talk about DNA structure. So DNA structure. Now before going to DNA structure, what is DNA? DNA is a nucleic acid that carries genetic information. Now when we talk about DNA structure, we should remember two important things. That one, DNA is a macromolecule and it is unbranched. Now basically macromolecules are the very large molecules composed of the polymerization of smaller subunits called monomers. Now in this case, the monomeric unit is a nucleotide. Now the nucleotide is composed of a pentose sugar, a nitrogenous base and a phosphate group. Now when we talk about the unbranched structure, it is because the two linear polynucleotide chain forms the structure. Now what do I mean by polynucleotide chain? That I'll be discussing in the next slide. Now when we talk about the shape, they are circular in shape. It is generally present in prokaryotes or the cells without a nucleus. Or it is linear in shape, basically found in the eukaryotic cells. Now the next one that comes is a sugar, or also we call it as a pentose sugar. Now this shows 4 carbon atoms with an oxygen forming a ring. So this is the oxygen which is forming a ring, and there is a carbon atom present at the 5' end at the outside the ring. Now this is the ribose sugar. The ribose found in RNA and is a normal sugar with one oxygen atom attached to each carbon atom. And what happens to the deoxyribose is found in DNA, and it is a modified sugar lacking one oxygen atom and hence named deoxy. So there is a loss of oxygen. So we can call it as it is a pentose sugar composed of 5 carbon atom, in which 4 carbon atom along with oxygen forms a ring, and the 5th carbon atom is present outside the ring. Now we know that nitrogenous bases are of two types, purine and pyrimidine. Purine, very well known, an example is adenine. Now in this, if you can see, I have labeled it as 1, 2, 3, 4, 5, 6, 7, 8 and 9, in which at the 6th position there is an amino group. So we can also call adenine as 6-aminopurine. Now as we see the next structure, it has got an amino group at the 2nd position, whereas at the 6th position it has got an oxo group. This structure is a guanine structure. So this is also known as 2-amino-6-oxypurine. The next one is the pyrimidine, and this one is a very well known structure of a cytosine. So here if you see at the 2nd position it has got an oxo group, whereas at the 4th position it has got an amino group. So we can also call it as 2-oxy-4-aminopyrimidine. Now the next one shows oxo group at 2nd and 4th position, which is an uracil, and it forms 2,4-dioxypyrimidine. The next structure, if you see, it is quite similar to uracil, as it has got oxo group at 4th and 2nd position, but there is a methyl group at the 5th position, and this is thymine, also known as 2,4-dioxy-5-methylpyrimidine. So if you see the structure of thymine and uracil, we can say that thymine is a methylated uracil. Now as we saw that the blue box contains the purine and the yellow box contains the pyrimidine, and if you see the structure, the purine is a bigger, or you can say the larger size than the pyrimidine. And to remember it, you remember it as pyrimidine has cut, C-U-T. And for purine, it is Ag. Now, I have listed out here some of the difference between purine and pyrimidine, so it would be easier for you to understand and as well as remember. So we know both are heterocyclic compounds. Purine, as I already said, it has got a larger sized nitrogen containing biomolecules, and pyrimidine has a smaller sized. The purine consists of two rings. One is hexagonal benzene ring and pentagonal imidazole ring, whereas pyrimidine has only one ring, that is hexagonal benzene ring. Purine is 9-membrane and pyrimidine is 6-membrane. The fourth point is the position of the nitrogen group. So if you can see 1, 3, 7, 9, and pyrimidine it is only 1 and 3. So 1 and 3 is common in both of them. Now for the base pairs, purine consists of Ag, adenine and guanine, and pyrimidine consists of cut, that is cytosine, uracil and thymine. Now at the sixth point we see that purine occupies more space in DNA, obviously because of the size, and pyrimidine occupies a lesser space in DNA. Now we already know what is a nucleotide. So sugar plus nitrogenous base and phosphate group forms a nucleotide. Now how is the structure actually? The structure looks like this. It's an example of the adenine. So we have a nitrogenous base, which is an adenine, which is linked with a sugar by glycosidic linkage, and the sugar is again linked with the phosphate group by a phosphodiester bond here. But what if there is only a linkage between a sugar and a nitrogenous base? So it is called as a nucleoside. Now, how is the structure? The structure consists of a nitrogenous base and a sugar, forming a glycosidic linkage or a glycosidic bond. Now, if you see the structure, this is how the structure is arranged. I think it is quite confusing for you. I can make you understand. So the S, as you can see, it is the sugar, right? And there's a nitrogenous base like A, T, G, C with different color. So the A, that is adenine, bind with thymine with a double bond. Cytosine binds with guanine with a triple bond, and this shows the phosphate group. Now what happens? This is a polynucleotide chain 1, and this forms a polynucleotide chain 2. Now in this, each nucleotides are linked by phosphodiester bond. And then what happens? These two polynucleotide chain that I showed you, 1 and 2, are linked with each other by the hydrogen bond. If you can see the hydrogen bonds. So we can say that nucleotides joined with each other forms a polynucleotide chain in which the adjacent nucleotides are linked with each other with a phosphodiester linkage. And two nucleotide chain are held by hydrogen bonds where adenine binds with thymine and cytosine binds with guanine. So now let's come to the summary. What we learned today. We learned about DNA structure, which is a macromolecule, unbranched, having a monomeric unit called nucleotides, and shape, which is a circular and a linear shape. The nucleotide is composed of sugar, nitrogenous base and a phosphate group. Also, when there is a sugar and a nitrogenous base, it forms, the nucleosides. Now this is a structure, how the phosphodiester bond is formed and the glycosidic bond is formed. This is a purine and pyrimidine. We saw a lot of different between both of them, but the basic is purine consists of Ag and pyrimidine a cut. This is a structure how the two polynucleotide chain are linked together, forming a hydrogen bond. I hope this video is helpful to you guys. You may have any inquiry, and if you have any, please feel free to comment below. Thank you.